Welcome. Good evening to all of you. Today, we are going to talk about something called biological engineering. I'm going to tell you some stories. That's essentially how I planned this. And hopefully, by the end of these few stories, you would have a good idea as to what biological engineering is all about. Biological engineering is a reasonably comprehensive term which has come into vogue over the past some years, let us say. Whereas the field itself is rather old. If you even go back about 60, 70 years, you had a term called zymotechnology, which was used to look at enzyme conversions and so on and so forth. And therefore, this field has always given products of importance, of significance to humankind. Even if you go back in recent history, about 150 years, it has given something or the other, but not in a short period of time and then it goes on, nothing like that. It has been a consistent kind of thing. And then from zymotechnology came more of biochemical engineering, if you want to call it that. It was a combination of biology and chemical engineering toward processes. And at the same time, there were people working with genetic engineering, modifying the genes and so on and so forth. In the 1970s, maybe. And then that started becoming a field. Therefore, it's kind of an evolving thing. So the people who are working with genetics and things like that like the term biotechnology. And so let me tell you the old terms, at least starting with biotechnology or biochemical engineering. These are old terms limited to chemical engineering and biology put together. That's how it started many decades ago, maybe. The idea, research, and so on and so forth. There were a lot of biochemical engineering programs that were there, and so on and so forth. In fact, when I did my doctoral studies, it was in the field of biochemical engineering. Now I call it biological engineering. Then I mentioned that you had this, biotechnology. Let's talk about biotechnology. Biotechnology was this, had this connotation of genetic engineering aspects and so on and so forth. May or may not be engineering related. So that term came along. And then sometime later, you had this term bioprocess engineering, which is still being used. It is a focused aspect. It looks at processes, more of bioreactors, downstream processing kind of applications. And so that is a term that is still in use. There is another term which is called bioengineering, which is somewhat equivalent to biomedical engineering. These are all perceptions. Different people will have different perceptions. So if you talk to another older person, maybe who kind of grew up in this time, they may have their own views on this. These are my views, which seem to reflect how they are perceived. Therefore, let me tell you that. So bioengineering, which is current term biomedical engineering, is also used nowadays. That is more of engineering aspects related to the human body and related aspects, or anything to do with human body health and so on and so forth. That is seen as bioengineering. Biological engineering, as you will realize later, is more of an all-encompassing term in a certain sense. You need to note that all these draw from the biological sciences or the biosciences. That is the fundamental knowledge base from which we all draw things to apply and so on and so forth. So this is, I thought I'll kind of clarify these terms when we begin with it. So that you'll have some idea in your mind or some clarity in your mind to receive the other information. However, these divisions are all fine at the undergraduate level. I don't really have too much of a problem. Although people, can get very sensitive about calling one the other and so on and so forth. Even calling biotechnology as biochemical engineering or bioengineering as biotechnology, people get a little upset. But don't worry about it. I have a different view on that. I think they are artificial. The divisions are artificial and they make no sense to me for actual contributions, as will become very clear. And what we're going to do is see what I mean. I'm going to play you some videos first. Okay. I thought of first talking for about 30 minutes or so and then taking questions. I think that would be the best way to do it. But even instead of talking, I thought I'll play you the videos from a source that you can all access. The links are all there. So that way it'll be much more interesting. Before I play the video, I need to tell you that either me, the author, or NPTEL or IIT Madras, we do not endorse the products that are shown in the video or the organizations that are represented in the videos. They were chosen for their ability to teach well, for their didactic value in this particular context. With that, let me play the first video. Let me request our Karthik here to play the first video, which is on vaccine production, which is very timely. We are right in the middle of this crisis now, the COVID-19 crisis caused by SARS-CoV-2, the virus. So you'll see this video and then note what all are being done. Where does the vaccine come from, how it is produced, and so on and so forth. Then we'll talk a little further on that.
It's a simple gesture, one we make dozens of times for ourselves and our families to help prevent potentially life-threatening infectious diseases such as meningitis, pertussis, flu, polio. Vaccines work by preparing the immune system to better combat infection and prevent disease. They are biological products that result from a series of sophisticated and careful scientific processes that ensure the quality of each shot. Here's how vaccines are made. Production starts with growing viruses or bacteria in the lab. A wide range of parameters must be managed, like temperature, pH, oxygen rate, sterility, and homogeneity. The slightest variation in the culture media may compromise the production and cause delays. Depending on the vaccine, cell cultures take from 2 days to 3 months. From a small amount of original cell culture solution, the manufacturer can eventually produce millions of vaccine doses. The microorganisms are extracted from their environment through a process called purification, which eliminates any trace of culture media. We then inactivate the virus or bacteria. That means we eliminate its ability to cause disease. We disarm it. But we retain its ability to elicit a precautionary immune response from the body. The inactivated virus or bacteria is called a valence. A vaccine can include one or several valences to protect against one or multiple diseases in the same shot. Each valence must remain fully effective, and it must not interfere with the other valences. This careful, precise combination takes years of scientific research and development to perfect. Then, stabilizers or a preservative are added in extremely small quantities to help guarantee that the vaccine will remain stable, potent, and effective. Sometimes an adjuvant is also added to enhance the body's immune response to the vaccine. Certain vaccines are also freeze-dried into a powder to help further preserve the vaccine's efficacy. This is particularly important for vaccines that are intended to prevent outbreak-prone diseases like yellow fever, where having available stocks on hand over time is an important element to combating and containing disease. When it's time for the vaccine to be administered, it will be reconstituted by combining this powder with a diluent. The vaccine in liquid or freeze-dried form and diluents as needed are then filled into vials or syringes. When the filling phase is complete, each vial or syringe undergoes visual inspection. The the quality of the contents and the container are scrutinized by both the human eye and state-of-the-art digital surveillance technology. All along the production, Sanofi Pasteur performs hundreds of laboratory tests on each and every batch of vaccine to confirm quality. In addition, samples are taken from each batch and sent to public health authorities for testing as well. Only after the vaccines have successfully passed both sets of tests can distribution begin. There are no exceptions. If a batch does not meet the quality criteria, it will be destroyed. Throughout packaging and shipping, vaccines must be stored between 2 and 8 degrees Celsius. Cold rooms and temperature-controlled shipping solutions, such as refrigerated containers and isothermal packaging, protect the vaccines during each phase of transportation and storage. A temperature tracking device is included in each shipment. Sanofi Pasteur takes care to follow strict precautions throughout the entire manufacturing process to ensure the quality of our vaccines. Production takes place in absolutely sterile conditions. Even the atmosphere is purified. Continuous control along the production chain also ensures that raw materials, equipment and manufacturing processes, and the finished product are of the highest quality. In total, more than 70% of the time required to produce a vaccine is spent on these quality controls. Depending on the vaccine, we conduct up to 1,200 different tests throughout the process. For these reasons, the total time required to produce a vaccine ranges from 6 months, for the flu vaccine for example, to 36 months for combination vaccines. For Sanofi Pasteur, assuring a reliable supply of quality vaccines is an essential part of our mission. We've been committed to developing and delivering innovative vaccines to people around the world for more than 100 years. As a leading vaccine company, we are proud to contribute to protecting health and empowering life.
Okay, that must have given you an idea as to how vaccines are produced. And if you had looked at that a little closely, you would have seen the vessels in which they were produced. Right? There were those stainless steel vessels, which I guess are about a few thousands of liters capacity and so on and so forth. They're called bioreactors, by the way. So, if you look at what to produce, that information actually comes from from the biosciences aspects. The people need to understand the disease and so on and so forth, the molecules that are affected by it, which is currently being done for COVID-19, for SARS-CoV-2. And what actually needs to be produced needs to come from that field. And then how do you produce it is all an aspect of biological engineering. Note that I mentioned areas. I did not mention the person. It is entirely up to the person to go and pick up whatever is necessary. So that's why I said the barriers are artificial. The person just needs to go across these perceived barriers, pick up whatever is necessary, or work in a team. That's the best thing. The expertise from these various disciplines come together, contribute towards a goal, and that's what results in a good contribution. Now, let us look at some different angles to build up the story on biological engineering. I'm going to play 2 videos in quick succession. Let me first show you the name of the video. It's called "The World is Poorly Designed." Okay. From human disease, a viral disease in this case, we are going all the way to something that you typically relate to as design. Can you play the second video, please?
In 1989, Japan's Shinkansen bullet train had a problem. It was fast, really fast, like pushing 170 miles per hour fast. But every time it exited a tunnel, it was loud. The noise was coming from a variety of sources, but whenever a train sped into a tunnel, it pushed waves of atmospheric pressure through the other end. The air exited tunnels with a sonic boom that could be heard 400 meters away. In dense residential areas, that was a huge problem. So an engineering team was brought in to design a quieter, faster, and more efficient train. And they had one secret weapon. Eiji Nakatsu, the general manager of the technical development department, was a bird watcher.
Different components of the redesigned bullet train were based on different birds. Owls inspired the pantograph. That's the rig that connects the train to the electric wires above. Nakatsu modeled the redesign after their feathers, reducing noise by using the same serrations and curvature that allow them to silently swoop down to catch prey. The Adelie penguin, whose smooth body allows it to swim and slide effortlessly, inspired the pantograph's supporting shaft, redesigned for lower wind resistance. And perhaps most notable of all was the kingfisher.
The kingfisher is a bird that dives into water to catch its prey. The unique shape of its beak allows it to do that while barely making a splash. Nakatsu took that shape to the design table. The team shot bullets shaped like different train nose models down a pipe to measure pressure waves and dropped them into water to measure the splash size. In 1989, Japan's Shinkansen bullet train had a problem. It was fast, really fast, like pushing 170 miles per hour fast. But every time it exited a tunnel, it was loud. The noise was coming from a variety of sources, but whenever a train sped into a tunnel, it pushed waves of atmospheric pressure through the other end. The air exited tunnels with a sonic boom that could be heard 400 meters away. In dense residential areas, that was a huge problem. So an engineering team was brought in to design a quieter, faster, and more efficient train. And they had one secret weapon. Eiji Nakatsu, the general manager of the technical development department, was a bird watcher.
Different components of the redesigned bullet train were based on different birds. Owls inspired the pantograph. That's the rig that connects the train to the electric wires above. Nakatsu modeled the redesign after their feathers, reducing noise by using the same serrations and curvature that allow them to silently swoop down to catch prey. The Adelie penguin, whose smooth body allows it to swim and slide effortlessly, inspired the pantograph's supporting shaft, redesigned for lower wind resistance. And perhaps most notable of all was the kingfisher.
The kingfisher is a bird that dives into water to catch its prey. The unique shape of its beak allows it to do that while barely making a splash. Nakatsu took that shape to the design table. The team shot bullets shaped like different train nose models down a pipe to measure pressure waves and dropped them into water to measure the splash size. The quietest nose design was the one modeled most closely after the kingfisher's beak. When the redesign debuted in 1997, it was 10% faster, used 15% less electricity, and stayed under the 70 decibel noise limit in residential areas. And it did all that with the wings of an owl, the belly of a penguin, and the nose of a kingfisher. There's a name for design like this. It's called biomimicry.
The people who design our world usually never take a biology class. Believe it or not, So they're novices in how the world works.
That's Janine Benyus. Back in 1997, she wrote the book that coined the term biomimicry. It told the story of the innovations in computing, energy, and health that were inspired by structures in the natural world.
Stick like a gecko, compute like a cell, even run a business like a redwood forest.
Benyus has since worked as a consultant for various companies, trying to get them to understand how to take design ideas from nature. That might mean studying prairie dog burrows to build better air ventilation systems, mimicking shark skin to create bacteria-resistant plastic surfaces for hospitals, or arranging wind turbines in the same drag-reducing pattern that schools of fish swim in. Designers get inspiration from a lot of different places, but Benyus thinks many of them could benefit from looking more at the natural world.
So there's a lot of sort of looking at what other people have done, and what they do is they look at all the others, and they get ideas. They literally do, a lot of designers have lots of magazines that they look through. They tear those out and they put them on inspiration boards. But they're looking at other human technologies.
Her idea was simple. Designers should get in the habit of bringing a biologist to the table and let them help solve problems by mimicking nature. And there are three main ways they can do that.
You can mimic its form or its shape. You might create a paint for a building that when it dries, it's got the same structure as self-cleaning leaves. Lotus leaves are notoriously great. They let rainwater clean the leaf because they have these bumps, and the rainwater balls up on the bumps and then it pearls away the dirt. So that lotus effect is physical, and you can create a physical structure on the outside of any product. Imagine that on the outside of your car. Rainwater would clean your car. So that's mimicking form. But there's also a mimicking process, the processes in the natural world. It might even be, how you mimic how ants communicate in order to efficiently find sources of food or new places to live. And those processes, that self-organization, has been mimicked in software. In things like autonomous cars and how they're going to move in flocks through the city by talking to one another. That's mimicking nature's process. And then you jump up to the level of mimicking whole ecosystems, right? There's a thing that's a buzzword right now that's really hot called the circular economy, which is essentially, industry saying, there should be no such thing as a byproduct in our manufacturing facility that goes to landfill. It should be used by something else, right? And at the end of a product's life, that product should be upcycled into something else. It's being called the circular economy. Ecosystems do that really, really, really well. You've got a log on the forest floor, and those materials move up into the body of the fungus that eats it. Those materials move up into a mouse, and that mouse material moves up into a hawk, right? And if you think about that as what we'd like to do with local materials being upcycled constantly in our cities, for instance, those ecosystem lessons are really big for us.
And that's the end goal for biomimetic design, making products, systems, and cities functionally indistinguishable from the natural world. Life has been around on Earth for 3.8 billion years, and what designers are starting to realize is that is a lot of research and development time.
The people who design our world have a lot to learn from the natural world. All they have to do is take a look.
Thank you so much for watching. This is one of a series of videos that we're doing in collaboration with 99% Invisible. They are a podcast that does stories all about design. We loved working with them. You should definitely check them out at 99pi.org or on any podcast app.
Biomimicry is innovation inspired by nature. It's the process of looking at something like a leaf and trying to figure out how to make a better solar cell. It's become popular in the design disciplines, mainly I think because people are looking for more sustainable ways to do things. And organisms know how to do this. After 3.8 billion years, life has learned what works and what's appropriate on the planet. And right now, that's what the people trying to redesign our world are looking for. What we do in biomimicry is we bring in biologists to the design table. We look at how does nature contain liquids? How does nature repel water? So for instance, go outside, look at any leaf and the veins in a leaf, and what you're seeing is the world's best water distribution network. There's an amazing thing called the Murray's Law that says that all branching structures in the natural world, including our lungs, they all follow a single mathematical formula. And it has to do with the pipe branches, and it drops down to a smaller diameter, and then it branches again, and that drops down to a smaller diameter, and that's predictable. People in green building now are starting to say, well, maybe our 90-degree angles that we have in plumbing are really friction devices. Maybe we should distribute electricity differently in a building, water differently in a building, even gases, even air conditioning differently in a building by mimicking this Murray's Law in our plumbing. The most important thing that people should know is that a sustainable world already exists. We're just now beginning to open our eyes and realize that the answers to the questions we've been asking, how do we live here sustainably, are all around us.
मेरा नाम शांता महतो।
Okay, fine. Good. Okay, I'm sorry that you had to face some technical issue. I was told that the video froze. It was fine here. Sorry about that. So these two videos, which the world is poorly designed and biomimicry, these eloquently told you the relationship between the various different aspects and the way they need to come together for better and better products, processes, and so on and so forth. Let's move further. If you look at bioprocess, I did mention bioprocess engineering earlier. The vaccine production was a bioprocess. If you look at the overview of it, it has two major aspects. One, the bioreactor aspect, which is something like that. It is an instrumented and a controlled vessel in which microorganisms grow and produce the product of interest, or they could be an enzyme there. The second major aspect is the downstream processing aspect. And I'm going to represent this by a conceptual rectangle there. That could have very many steps. That could have about 95% of the steps in an entire bioprocess. So the raw material gets into the bioreactor, and there it gets converted by the microorganisms. The microorganisms need to first grow, in other words, multiply, using the nutrients and so on and so forth, or an enzyme to do the conversion. And what comes out is, in that stream, you find product, the microorganisms themselves, and many other materials. Our interest is obviously in the product alone, as pure as possible. And so we go through downstream processing steps to get the final purified product. And that process is called harvesting. We don't have to worry too much about it. To get the final purified product. That is what a bioprocess is all about. All the details associated with it go into bioprocess engineering. And to tell you another story, we all know insulin. We all know how important insulin is in managing diabetes, right? And that's a widespread health condition nowadays. Insulin was discovered in 1920s, by the way. Okay, it's been around for quite a while, and it was obtained from pig pancreas till about the 1980s. Pigs, pancreas is a small organ in a pig, and that was the source. And so you can imagine what it would have been like. 800 to 1000 kg of pancreas is needed to produce 100 grams of insulin. Those are the numbers there. Whereas, and of course, pancreas, as I mentioned, is one of the small organs in the body. And once a bioprocess was found for insulin, it took away all this and immediately, very soon, reduced the cost by about 24-fold. There was a totally different way of producing insulin. And that was much better in many ways. I think I will play the last video also. This is a nice story. So with this video, we'll get back to some talk and then I'll take some questions. Okay. Can you play the next video, please? Thank you.
Almost 1 in 10 adults worldwide has a condition known as diabetes. And many of these 400 million people rely on life-saving injections of insulin. But for nearly 50 years, the only insulin available came from cows and pigs. Then a Danish company worked out how to make synthetic human insulin using the key ingredient in a loaf of bread, baker's yeast. So, how do they do it? Welcome to Novo Nordisk in Kalundborg, Denmark. This vast plant covers over 1.3 million square meters, making it twice the size of the Pentagon, the world's largest office building. It needs to be big because they produce almost half the world's supply of synthetic human insulin. Remarkably, they make it using the same stuff that gives rise to your loaf of bread. Dr. Jens Kierkegaard Beck explains.
To make a good bread, you need the yeast. The very same yeast is also used for producing the insulin, saving millions of people's lives.
As Jana Futlov demonstrates just for us, all it takes is a tiny amount of yeast to produce enough insulin to treat thousands of diabetics. 1 milliliter in a vial like this can generate enough to save up to 500,000 lives. But this is no ordinary insulin. It's been cleverly genetically engineered by adding the gene for insulin production to its DNA, turning it into a super yeast. The next problem is, yeast is a living microorganism. It needs feeding before it can produce insulin. The solution is a solution. A nourishing mixture of water, sugars, salts, amino acids, vitamins and minerals. Once it's got all the nutrients it needs and it's warmed up to a cozy 30 degrees Celsius, the yeast does what comes naturally. It starts feeding and fermenting, producing CO2. But thanks to its modified DNA, it also does something rather unusual. It secretes insulin molecules. At Kalundborg, they've taken the process that Jana demonstrated in a glass flask and repeated it on an industrial scale in this massive 200-meter-long production hall. The modified yeast is added to large sterilized stainless steel flasks containing generous quantities of water, nutrients and sugar. Warmed to 30 degrees, the resulting fermentation broth is left for 72 hours for the yeast to go to work. The problem is, after 3 days, the yeast has produced just a few hundred grams of life-saving insulin, and that's mixed in with tens of thousands of litres of this murky broth. So they need to purify it. Purification begins with a spin in a centrifuge, filtration, and a 24-hour stint inside a capture column.
In this capture column, we go from this solution to a clay, a grayish-like substance containing 80 to 90% insulin.
After a second round of purification, the insulin needs to be dried. But there's a problem. Most dryers use heat. But the precious insulin degrades under warm temperatures. So they move it to this sci-fi drying room where it dries at just above freezing for 24 hours. Now it needs to be bottled before being dispatched to doctors and patients. But the bottling facility at this plant looks like it's from another dimension. If Stanley Kubrick ran a factory line, this would be it. They start with powdered insulin. A typical dose weighs just 1/1000th of a gram. Incredibly, this flask contains enough to treat one person for over 2,700 years. But before anyone can use it, it has to be diluted with distilled water. Then sent down the line, ready to fill thousands of glass vials. To ensure they're contaminant-free, the vials are washed, dried and sterilised at around 300 degrees Celsius. Insulin needs to be dispensed with an accuracy of millionths of a gram, so the vials are filled with computer-controlled precision. They're then fitted with an aluminium cap before passing through a rigorous scan and cam check. The final question is how to administer the insulin to diabetics. The answer is with something called a pen. But you wouldn't write with this. It's a needle and distribution system in a handy plastic tube. It takes less than 2 minutes to construct each plastic pen around its glass vial. And
Okay, you would have realized that all the insulin comes from a genetically engineered organism, bacterium, right? So that's pretty much the source now, and that's what is being used all over the world now. Let me tell you a few more stories, and I think I need to speed up now. I'll tell you a few more stories and then take questions. You see this? I'm not able to read anything. I don't expect you to read anything. This is some small fraction of the reactions that are happening in a single biological cell. Okay? There are thousands and thousands and thousands of reactions. If you go to the age-old biological processes of simple thing, curd making, you know why milk becomes curd? What do? You add a little bit of old curd and you set it aside at a certain heat to a certain temperature, set it aside, and then it becomes curd. And if you look at one set of reactions there, let me see whether I'm able to point out. If you look at the center, you see some molecules in red and so on. Yeah. Right here. Okay, and here, somewhere here, you'd find some pyruvate, I think. I'm not able to make out. So from pyruvate, you have a side branch that goes to lactate, and lactate is lactic acid that comes out of the cell that decreases the pH, coagulates milk, and that's how curd is formed. And acid adds flavor, adds acidity to curd, and so on and so forth. So that's a simple example. Another interesting example is the same pathway. Many cells produce ethanol in the place of lactate, right? And that is pretty much the source of all ethanol, whether it be for solvent, for recreational purposes, and so on and so forth. That is the source. Okay? So any byproduct is a part of those reactions that occur there, or the catalysts that make those reactions happen, called enzymes. Okay? So this is mind-boggling, the number of reactions in the cell. I just thought I'd show it to you. That is a source from which I took that. So there are thousands of reactions that take place in the cell all the time, and they can be viewed as a network of reactions. Okay? So we have a certain analysis framework to look at them. And the networks could either be the metabolic networks, the reactions networks, or genetic networks. The genetic information is what is used to make the proteins or the enzymes which catalyze these reactions, metabolic reactions, and so on and so forth. There could be various different networks. And these networks could be seen as producing the products of interest. And therefore, if you want to improve the production, this is where you need to look at the network of these various metabolites, genes, and so on and so forth. Okay. So we need to make sense of the inner workings of the cell. How do you make sense of the inner workings of the cell? Right? The inner workings of the cell could give us products, could help us treat or manage very difficult diseases such as cancer, Alzheimer's, cardiovascular, and so on and so forth. Many diseases for that matter. They could provide prosthesis. For example, if a person loses eyesight by some difficult means, let's say the retinal cells are gone, okay, there are prostheses that can help recover the vision for the person at a fundamental, if you understand it at a fundamental level. Therefore, understanding these aspects is a very important part of biological engineering. Analysis of these networks so that you get insights and you come up with ideas to manipulate them towards better and better products, processes, better control of diseases, and better prosthesis made. Okay, so this would have given you some idea as to what biological engineering is all about. And now I'm going to tell you how we get a graduate up to speed in these aspects, okay, how we go about doing these things. Not many people know about this. And what I'm going to tell you now is, in fact, common to any engineering curriculum. The engineering undergraduates in the respective disciplines are first given the knowledge, okay, information, knowledge, and so on, and are helped to understand the same toward analysis, design of those appropriate systems, and operation after graduation. Okay. That's pretty much what engineering is all about. Okay, we give you the knowledge, then we help you understand it. Then you need to analyze the systems. You need to, so that you can design better systems or operate the systems, troubleshoot existing systems, and so on and so forth, and contribute to better products, processes, and so on. For example, mechanical engineers are expected to analyze, design, and operate mechanical systems. Electrical engineers are expected to analyze, design, and operate electrical systems. Chemical engineers for chemical systems, and so on and so forth. Any engineering you take will fit into this framework. Similarly, biological engineering, okay, now it becomes a more comprehensive term. Biological engineering graduates are expected to analyze, design, and operate biological systems. And for the above, they need to have an appropriate understanding based on the suitable knowledge provided to them of the fundamental unit of life, which is the cell. To achieve this, we do something like this. First, we make sure that your base in the sciences is as it needs to be to build on, as strong as it needs to be to build on. Typically, students come with some reasonable levels in mathematics, physics, and chemistry because of the various exams they write, the 12th standard preparations, and so on. Whereas biology, they are not prepared because many of them don't even go through this. I've done this survey in our classes. About 90% of students or 95% of taught entire engineering, many engineering disciplines together. The Introduction to Life Sciences course, I've done this survey pretty much every year for many years. About 98% of the students have not done biology in their 12th standard. Whereas biology is essential. And therefore, we give them a full complement of basic biology courses at this stage. Microbiology, biochemistry, molecular biology, cell biology, and so on and so forth, because it is essential. We spend a lot of time doing that. Other engineering disciplines don't need to do this because they pretty much have it set up. Whereas for us, it is essential, so we spend the time in giving this full complement of the fundamental courses. Maybe later, when the world realizes that biology is important enough and people come prepared in biology, we can start at a different level. But right now, we train them in biology. On top of this base, you have pillars of analysis. I can think of 4 pillars: thermodynamics, both the classical aspects and the statistical aspects at an undergraduate level for biological engineering, both are essential. Transport aspects, movement, and so on and so forth, movement of physical quantities. Cell mechanics is a very essential pillar. And nowadays, data sciences, dealing with large sets of data. You know that DNA, as it is called, you may not know what DNA is, but you might have definitely heard that DNA has about 3 billion base pairs. Okay. You're talking of billions there. So you need to have means of looking at that. These are the pillars of analyses of biological systems. And on top of that, we have courses that give them further analysis and information, courses on design, lab courses which help them pick a lot of skills, necessary skills. And then we have electives. Electives, they could be of various kinds. They could take the analysis further, or they could give new information. The new information ones are reasonably short term, maybe for 5 to 10 years, maybe 15 years, and so on and so forth. They keep changing, they're topical, they keep changing with time. Whereas these courses, the pillars of analysis, they would stand the test of time, 50 years plus. Okay, unlikely that anything major is going to change. Of course, things like data science is coming in because the field itself is evolving as we speak. It's a new field, it's evolving as we speak. Those things would happen, but otherwise they have a long, their relevance is very, very long. So you have electives, you have humanities. We take students between 17 to 22, 21, and we need a full complement of humanities. I've just placed it there for this particular discussion. It's one of the basal aspects to have for a well-rounded undergraduate to humanities courses. Then you have a project. And then the person who comes out is a well-rounded graduate capable of analysis and design of biological systems or biosystems. So this is the thinking behind the curriculum. Not many people know about this, and that leads to a lot of other difficulties. This is the basis. And fine, having a graduate is fine, but what will the graduate do after graduation? There are 2 major paths. One is the industry, startup, or joining an established industry. Nowadays, startups are popular among people. A lot of people are getting into startups. And biological engineering is prime for getting into startups. There are a lot of contributions to be made, a lot of money to be made also, once you hit it right. But of course, you need to be oriented that way. And if you're oriented that way, then that's a good line to take. Otherwise, there's an established industry. Or people get into higher studies. After higher studies, they either get into academia or to the industry at a higher level, and so on and so forth. The industry typically looks at short-term aspects for day-to-day needs. Okay, they have vision and all that, but they look at short-term aspects. That is their relevance. Whereas academia looks at long-term aspects on a day-to-day basis. That's their relevance. Okay, that's its strength. Yeah, I think. This is the last thing that I wanted to mention. There are 2 aspects. Okay. Each person is different. Okay. I'm different from somebody else, and so on and so forth. And that's the reason why you need to choose on your own depending on your understanding of your strengths. There is an aspect of interest, whether something excites you, you're drawn to it, and so on and so forth. Okay, that's interest aspect. Then there is an aspect of abilities, okay, which is how effective you are in performing it. There are 2 different aspects. I might have a big interest in music, whereas if I start singing, people will run away. Okay, so I don't have any, I'm not effective in performing it. So both these need to come together. And when you make a choice, it's best to make a choice with these 2 coming together. So once you make the choice, then you'll have to work hard on very many different aspects. There are some things that will come naturally to you. There are some things that you'll have to work hard. But when you make a choice, you need to make a choice based on these two aspects. I thought it will be good for people who are at the crossroads to know this. I look forward to seeing people with interest and abilities in biological engineering in the various programs in biological engineering in the country. Hopefully that gave you some picture of biological engineering through stories and so on and so forth. And I think this is the right time to take up questions. Yeah, I went a little far, but yeah, let me take up questions now. Can you switch me to questions, please, sir? You can increase the size, that will be great. Okay, I can just about read it, I think. 150, 200 maybe, yeah. No, that doesn't change that changes only this. That's okay. Fine, I'll make do with this. Let's not spend too much time on that. How biological engineering can be used in the medical field. We saw very many examples, I think. This was asked before the thing began. That's okay. The biological fundamental through, can we know more in depth about coronavirus? Yes, okay, but a lot is not known about the it's new. It's new, and so people are racing against time to know more about it and come up with a vaccine and so on and so forth. Yes, it's certainly very much in the area. I'm mechanical engineer. Tell me the scope. Yeah, we did look at various different aspects of relevance of these two mechanical engineering aspects, or where you can bring in the skills in mechanical engineering to contribute to improving a biological system. Okay, I need to wait. Then difference between, yeah, this we covered in detail. Difference between biotechnology, bioengineering, biosciences. Yeah, first slide. The next question is, can an MBBS student get into the field of biological engineering? If so, what is the path she should choose? We have had our senior students, our PhD students from the MBBS field. We've had a few medical doctors who have done doctoral research in the field of biological engineering, biosciences, and so on and so forth. So that's very much possible. Definitely in IIT Madras, definitely in many other places too. Please explore. Then what is the scope of electronic engineering? Sky is the limit in terms of your imagination and so on. So there's so many applications of electronics engineering, all the sensors aspects. Any communication aspect is all electronics, whole host of applications of electronics. Can I look at what is the scope of the field in the future? I think this is the field of the future. I need to tell you something. When the undergraduate programs were kind of suddenly started, this was a top-down approach from the government. Government wanted us to start this sometime in 2001, 2002. IIT Madras started its program in 2002. It was anchored in chemical engineering at that time, given the way the field was developing. Then in a couple of years, we became an independent department, a separate department, independent department. What is the question again? I'm sorry.
Sir, can I understand the biology engineering, do master in robotics?
Just before that. Why did I get into that?
What is scope of?
What's the scope of the field in the future? So at that time, there was this big hype about IT. Okay, IT gave results so quickly, and BT is expected was supposed to be a big wave and so on. So, but we were all saying that it will certainly give very significant contributions. Certainly make significant contributions. It has made throughout history. If you read history of biological engineering, you'll see this. But it's not going to be as quick as IT is, and that's what it has turned out to be. It will certainly be relevant for a very long period. Next question. Can an undergraduate in biomedical engineering do masters in robotics? I don't see a problem. If you might need to pick up the necessary background. Okay, crossing fields is not a big deal. It's just getting out of your zone of comfort. Okay? You might need to pick up something. So if you don't have a flair for mathematics and so on and so forth, that's a different aspect. You need to choose things that are suitable. Whereas if you are an engineer and if you've done reasonably well in mathematics, it's just a choice. You just go. Of course, you need to work harder, pick it up, and so on and so forth. I don't see a problem with that at all. Just want to know how to register for the webinar. E-certificate. I don't think e-certificate is given for the webinar, right? Yeah. Okay. Then any other? These are comments. Thank you. Thank you very much for your kind comments.
So where are the questions?
That's it, is it?
Here is the question.
Here are the questions. I'm pursuing masters in biotechnology. What are the opportunities can see in bioprocessing now? Oh, the IIT Madras has a M.Tech in bioprocess engineering, okay, which is quite a recent program. The first batch would be graduating now. It's a beautiful program. If you are interested in bioprocessing and if you're looking at an M.Tech, definitely you should consider that. Then, I mean, I'm sure there are programs in other places also in bioprocess. Bioprocess is the traditional one. It will certainly be there. Even if people call it something else, biotechnology and so on and so forth, this would be there. Of course, make sure that it is there. Make sure that the courses in bioreactors, downstream processing, controls and things like that are there. And go ahead, you fit in. Any other questions?
Yes, sir. What are the best company to join?
What are the best company to join? There are a lot of good companies, is the best that I can say in a public forum. There are a lot of very good companies which have done great work, made good contributions.
Why is genetic engineering still underrated in India?
I don't think it's underrated at all. It's big. Of course, there is there is something that you need to understand. Sometimes there are social issues, okay, which are important, and only if you overcome those social issues would the contribution reach the people. And genetic engineering is a classic example of that. Okay. There are well laid out ways by which you need to do these trials and so on and so forth. As long as you do them, they are safe. Safe as okay. Let us take a parallel here. Okay, you have a variety of cars, and unfortunately there are accidents that happen. So if you argue that there should be no cars, there's going to be a problem. But at the same time, you need to be careful. You cannot risk a major catastrophe. That you need to be careful. But many of these are, in terms of genetic engineering, are properly tested and so on and so forth. So if they are done that way, then I think the benefit should reach the people. I already told you an example. All the insulin is made by genetic engineering too. Not many people know this. People happily inject themselves with insulin. All that is from genetic engineering. Any other questions?
So what will be the scope for the
biochemical engineer, and what will be the trending areas in which one can make our future? And what I would it's nice to be aware of trending areas and so on and so forth, and it's rather difficult to predict. Okay? That's like predicting whether a movie would be a hit or not. Okay? That's as straightforward as that. There are various different fields that are coming up right now, and depending on the need of the society, they would be a hit or they'll take their time. Okay. Just see, hit means it's popular. People talk about it and so on and so forth. It doesn't mean it's the most important thing. And a simple example is a video that goes viral. It's viral for a few days and that's it. Right? You don't get to hear of it later. It had its play. It had its fixation in the human mind for some time. Okay. That is what this trending is all about. I would go more by what are your strengths and where you can contribute. And there are contributions that you can make right through. If you want popularity for those contributions, that's a totally different thing. I don't think it's that important.
Please share some good project topics going on this field.
There are project topics. Project topics are dependent on the institution that you're in. The each institution has a certain flavor to it, has certain strengths associated with it, and projects in those areas would go on. Any area, I think, is fine. Can a mechanical engineering teacher with a batch of students near premises? I need to check with the dean. There are well-written guidelines. I think an MOU is required between institutions, and if there is an MOU, I think it is allowed. Of course, we're not talking of right now. Right now, nothing's happening. The institution is closed. Whereas in regular times, there are a lot of these requirements that need to be met for, and even then I'm not too sure of the projects. You see these, I used to be in touch with the ever-changing set of rules at one point in time. I'm no longer in touch with what changes very quickly. These things change, the guidelines change rather quickly. If I remember the guidelines right, the MOU I remember certainly, there has to be an MOU between the institutions, and then they were allowed. I need to check whether there's been a change to it.
Bioengineering in agriculture.
Oh, huge. Bioengineering in agriculture is huge. Instead of human being, it's a plant that you need to focus your interest on. Everything that we talked about with the human angle are applicable to plants also. And plants are the ones that provide us with food and so on and so forth. So very important, definitely. There's been a lot of contributions by the biological engineers, what are now called biological engineers. They're called by other names earlier, to the field of agriculture. Certainly, genetic engineering has made major contributions in agriculture. All your pest resistance, all your various different strengths in crops were brought about by certain genetic engineering. The process of grafting itself is in principle. Yeah.
So I would like to especially.
Yes. I would especially like to know that the industry or higher education institutes.
Like in a student experience.
Student experience. I'm unable to understand the question. If you can rephrase the question and send it to us, that will be nice. I'll try to answer that. What's the time now? It's when do we need to stop? We can
last question.
Last question. Okay.
Myself from
MBBS graduate. Can I pursue biomedical engineering? And what course? Higher degrees, yes. Undergraduate, you need to check whether you want an undergraduate at all in biomedical engineering or MTech in biomedical engineering with MBBS. PhD, I think we have had MBBS students do PhD in our department in biosciences, biological engineering, and so on and so forth. Our department definitely is. We also have biomedical engineering program in applied mechanics, and we have this work going on in a few departments in IIT. I need to check with their current requirements and so on. I don't think that's a problem. I think. Okay, thank you for your attention. Hopefully you gained something about biological engineering. You got an idea as to what biological engineering is all about through stories. See you then. All the best to you.